Hi, welcome everybody. Thank you all so much for joining our open stage webinar. This is a series that we do from the Office of Programs to Enhance the Neuroscience Workforce within the National Institute of Neurological Disorders and Stroke. We're very pleased to welcome you. This is kind of our targeted outreach effort that we do for our diverse neuroscience community to learn about NINDS programs and network with NIH and NINDS program staff, and also the people funded by NINDS. So before we get started, I just have a few housekeeping items. So all attendees are muted, so please stay on mute. You will be able to ask questions throughout the webinar, so please use the chat to ask any questions. We're going to keep it kind of high level for the questions during the recorded first hour of the session. Then we're going to have an opportunity for all of you to meet the speakers in breakout rooms. So we're very excited about that. So if you have kind of specific questions for a speaker, please save those for the breakout rooms. You'll be able to move between rooms and meet all the speakers. So we're excited for you to have that opportunity. You can enable closed captions under live transcript at the bottom of your screen. As a reminder, this meeting is being recorded. It'll be transcribed and archived on our website. And I will also send a very long email at the end of this webinar, probably in a week or so, with the recording, with the slides from this webinar, and a bunch of resources that we share today. So don't worry about missing anything if you have to duck out or you didn't catch something. So we'll send you all the information soon. All right, please do stay connected with us. Follow us on Twitter, everything else. And we're so excited to have you here. I'll pass it off now to Dr. Devi Mahapatra to get us started. Thank you.
Okay, thank you, Anna. Can you hear me clear? Okay. So again, good afternoon and good morning, everyone, based on where you are, to this NINDS Open Stage webinar. And this is specifically on the NIH HEAL initiatives and more on the role of NINDS there in the NIH HEAL initiative. So I'm a program director in NINDS with a pain portfolio in the systems and cognitive neuroscience portfolio. So before I go through the HEAL programs and opportunities, which will be followed by presentations by my NIH colleagues and grantees on workforce efforts, pain therapeutics development efforts. First, I would like to start by briefly touching on pain as a public health crisis point. So as almost all of us here are very much aware that in addition to opioid epidemic, we are going through another public health crisis, that is pain. Pain is the most common reason behind people seeking medical care, with more than 50 million Americans suffering from chronic pain, which interferes with multiple aspects of our daily life, and it comes with a tremendous economic burden. And chronic pain affects more rural population than urban dwellers, as well as more in women than men who face chronic pain challenges. In fact, there are more new cases of chronic pain compared to other major chronic health complications such as diabetes, high blood pressure, and depression. So from NIH side, NINDS has been the lead institute for pain research, supporting basic translational and clinical pain research to develop new and innovative advances in the field of pain. And NINDS mission of pain covers a wide range of pain conditions. And as you can see, some of the examples shown here on NINDS supported research in pain that has led to discoveries of molecular, cellular, circuit, and system level discoveries and understanding of pain biology at both peripheral as well as central nervous system levels. In fact, the 2021 Nobel Prize in Physiology of Medicine was awarded jointly to Dr. David Julius and Dr. Ardem Pataputian for their discoveries of receptors for temperature and touch that are highly critical for the detection and perception of pain. Also, the 2021 Brain Prize was awarded to Dr. Michael Moskowitz, whose NINDS-supported research showed that a migraine attack is triggered when trigeminal nerve fibers release neuropeptides such as CGRP or the calcitonin gene-related peptide. And in fact, his research proposed that blocking the action of these released neuropeptides could be a new approach to treat migraine, and which has in fact led to several clinically approved and used therapeutics for migraine successfully. NINDS supported research has led to 2 FDA-approved pain therapeutics in more recent past, and several new analgesic therapeutic agents, as well as nerve stimulation and neuromodulation devices, are currently at different stages of clinical trial. And NINDS, alongside NIAMS and NCCIH, leads the NIH HEAL initiative's efforts to enhance pain management. And also NIH leads the executive committee of the NIH Pain Consortium, which includes almost 21 NIH institute centers and offices who are involved in pain research, supporting pain research in multiple ways. Going a little bit on the brief overview of NIH HEAL initiatives, pain research goals and priorities. So it aims to actually reduce pain and the risk of opioid use disorder by mainly developing safe and effective pain treatment and prevention strategies to improve quality of life for all people. So it covers every aspect of pain, from very basic biological understanding of human pain all the way to non-addictive pain treatment developments, different pain modalities for treatments, as well as best practices for effective pain management. So far, since 2018-2019, the NIH HEAL initiative has invested over $1.2 billion on pain and cross-cutting research, and looking into the programs where these investments have been made, and these investments are still continuing. So yeah, you can see here, it's a snapshot of all programs. It's a very busy and complex slide. My apologies for this. But these are the complex but very well organized programs developed and implemented through NIH HEAL Pain programs. And you can see there are 3 major categories of these programs. One more towards discovery or understanding biology of pain, specifically on human pain and pain conditions, to non-addictive therapeutic, specifically analgesic therapeutic development, to clinical development trials and effectiveness research. So these individual programs in these 3 major pain domains are depicted here in individual rectangles. And the asterisks here shown here are these programs where NINDS is the lead. Also, you can see in the rectangle at the bottom and of the entire chart here, which is on the workforce development and diversification in pain, on which you will hear more on subsequent slides, as well as in subsequent presentations. So one more important aspect, really important aspect in HEAL initiative is about diversification efforts in pain. So HEAL pain programs have really emphasized on inclusion and diversification plans for enhancing diverse perspectives in all grants, cooperative agreements, and research applications, where they are mainly like score driving criteria. And those, in fact, address 3 aspects of diversification and inclusivity. One, in the research workforce, second, in the study participants, and third, in the research and our clinical professional environment. So how all these 3 together really could enhance the diversification and inclusivity in pain research. So furthermore, NIH HEAL initiative and specifically the pain program on HEAL initiative has significantly widened or expanded the community engagement processes with multiple approaches and strategies in these efforts. So the community engagement with diverse groups and individual affected have been really strengthened by engagement at multiple levels, from research to clinical care, engaging everyone, but in utilizing different strategies through work groups, through workshops. So this has really expanded and brought in the absolutely important aspect of pain research and clinical care, that's feedback and input from people with lived experience. And another aspect which is really important in HEAL initiative, I would like to cover here a little bit, is the effort on health equity and pain management. And these programs are developing really research interventions to reduce disparities in pain, and mainly studies that are focused on populations that experience health disparities, including minoritized racial ethnic populations, rural populations, sex and gender minorities, persons with disabilities, and those with low socioeconomic status. So these studies also have a requirement to have a detailed community engagement plan that really outlined how they would be involving people who are with lived experiences, not just at the beginning, but throughout the entire study. So in continuation, HEAL initiative is really highly interested in understanding factors that contribute to health disparities in pain, specifically the stigma, bias, and/or discrimination at the patient level, at provider level, at community and/or system level, that really contribute to disparities in pain assessment, pain treatment, and pain management. Also patient-related, healthcare provider-related, and healthcare system-related factors that really contribute to disparities in pain assessment, treatment, and management, as well as factors such as social determinants of health that deter really engagement of patients across clinical pain management spectrums. And so also the barriers to really quality pain prevention, pain assessment, as well as treatment and management. So with this, I would really like to hand over the presentation to Dr. Laura Warner to talk on HEAL Pain workforce efforts. So Dr. Warner, please take it over.
Thank you so much, DP. I really appreciate it. Well, good afternoon, everyone. My name is Laura Wandner, and I'm also a program director in the Office of Pain Policy and Planning at NINDS. And I'm going to spend some time talking about the HEAL Pain workforce efforts. And as DP was just mentioning, he was explaining the overarching HEAL initiative. And oops, sorry, we skipped a slide there. HEAL research is really helping to try and improve pain management, and the HEAL research efforts cross a variety of areas of research. The one that we're going to be spending some time talking about in my talk is training and mentoring, and really that HEAL has been spending a lot of time trying to find ways to enhance the research workforce. And so the reason why we're trying to work on enhancing the pain research workforce is that we know that at all career stages that there are different ways and reasons why people are dropping out of the workforce. And that could be limited pain education about pain. It could be lack of mentors, protected time. There are lots of reasons. And although that this figure here is for clinical pain research, we know that these efforts and these problems span not only clinical, but also preclinical and translational research. So as a result, the HEAL initiative is really seeing this workforce crisis as an all hands on deck opportunity that we really must ensure an expanded and enduring diverse pool of highly trained scientists to perform high quality pain research. And so on the right hand side, you'll see some grants that the HEAL initiative has already issued to help try and increase the pain workforce. And we'll talk a little bit more about these opportunities over the course of this talk, but also more than happy to answer any questions you have in our breakout sessions. So recently, HEAL has established a pain workforce work group, and it has a very long mission. It's actually 2 slides long, so please bear with me here. But I do think it gives sense of really what we are trying to work on within the HEAL initiative. We're trying to cultivate a diverse and innovative pain research workforce community that's capable of meeting the challenges of studying and treating people with lived pain experience and comorbid conditions from a biopsychosocial perspective. And we're really aiming to recruit and retain and educate both basic, translational, and clinical pain researchers from diverse backgrounds and from a variety of perspectives and skills. So the goals of our efforts are to provide workforce opportunities for pain researchers across the spectrum of pain research, to attract and retain talented pain researchers from diverse professional backgrounds, socioeconomic backgrounds, regions of the country, and to attract researchers from non-traditional pain research backgrounds. We're also committed to enhancing pain education or continuing education, supporting professional development of pain researchers across all career stages, foster collaborative and supportive community, and promote an inclusive culture from all backgrounds. And so by achieving these goals, the NIH HEAL Pain Workforce Group is hoping to enhance the capacity of pain research community to address the critical challenges of improving pain management care for those living with pain and identifying and developing new and effective non-opioid treatments to manage pain and ultimately improving patient care and outcomes. And the co-leads of this workforce work group are myself at NINDS and Steve Pittenger at NCATS. And we are looking to try and expand our HEAL workforce efforts. But what this figure shows here is that between 2021 and 2024, there have been 4 total awards for workforce efforts. These awards have been made across 14 states, and the grants have been awarded to investigators at all career stages from postdoctoral trainees and through senior investigators. So both HEAL and NIH have recognized that there are workforce needs. So we've been trying to identify problems as well as how to address those problems and come up with solutions. So the first problem that was identified was a need to diversify the pain research workforce. So one solution has been to provide supplemental funding for HEAL awardees to recruit diverse individuals into their projects. So I'd encourage people who might be interested in this opportunity to monitor the HEAL website for future funding opportunities and to reach out to HEAL investigators to provide a supplement to their award. Another solution is to support small-scale research projects at educational institutions that provide baccalaureate or advanced degrees for a significant number of nations' research scientists, but have not been major recipients of NIH support. So this grant opportunity, it's called an R15. Again, happy to talk about some of these in more detail in our breakout session. Is really trying to promote diversity in the workforce. So if you're interested in this opportunity, again, please monitor the website, and the program leads for both of these are on the bottom of the slide that you can reach out to. Another problem that was identified is that there are not enough early career researchers to replace those leaving the field. So one solution is to provide supplements to train novice researchers and investigators new to pain by providing supplements. So again, I'd encourage people who are interested in these types of opportunities to monitor the HEAL website and to contact a HEAL investigator to ask to submit a supplement as part of their HEAL award. Another solution has been to release a national scholar grant where researchers will receive enhanced mentorship for 2 to 3 years. These are for early career researchers. And the aim is to fund researchers who are not yet ready for an independent NIH grant, so an R or Career Development Award. And these scholars cohorts are meant to be multidisciplinary and diverse. And so this opportunity is what we call the HEAL National K-12 Award. And it's being managed by the University of Michigan. So if you're interested in this award, please feel free to either reach out to the University of Michigan or to myself. And that scholar applications for the next cohort are going to be due in 2025 if you're interested. Continuing with that idea that there are not enough early career researchers to replace those leaving the field, another solution has been to increase the number of postdoctoral opportunities for clinical pain researchers. So the T90, R90 postdoctoral centers, this is a specific postdoctoral opportunity that we can talk about in the breakout session, are about to be funded. So we would encourage postdocs or people who are interested in applying for postdoctoral opportunities within the pain field to check the HEAL website to see if a postdoctoral training center was funded that they'd like to apply to. And each center will be sending out a call for postdoctoral applications sometime around the fall or winter of 2025. So if this is of interest, please keep your eyes out for this opportunity. And then another solution to increase the clinical workforce is that HEAL has the intention to fund career development awards such as K23s and K08s. So for clinicians who want to go into a field of clinical research. And we would encourage you to monitor the HEAL website for this funding opportunity. So if you're interested in either of these, please feel free to reach out to me since I'm the program contact. More than happy to answer any questions you have about them. Another opportunity here is the K99R00, which is an advanced postdoctoral to independent career transition award for both pain and substance use disorder. And the goal is to support a cohort of new and well-trained independent investigators from diverse backgrounds in order to promote a diverse pool of available independent investigators working in research areas supported by NIH and the HEAL initiative. So if you're interested in this initiative, we do have open dates through 2025, and the program contacts from NINDS are here listed on this slide. And another problem that has been identified is that there's a need to increase the contact with mentors, increase opportunities to collaborate with pain researchers across the spectrum of pain research, from preclinical, translational, clinical research, and to centralize resources and trainings. And so one of our solutions has been to establish a network to increase mentorship, career development throughout the year, and organize an annual meeting to communicate and facilitate communication across the spectrum of pain research. So this is what we call the Purpose Network, and the PI of the Purpose Network, Jacob Coverstone, will be speaking right after me and going in much more in depth about the Purpose Network. So stay tuned for that in just one moment. And another problem that we identified is that researchers have questions about grant mechanisms, open funding announcements, and just tips and tricks about how to write a strong grant. So one of our solutions was to run a monthly webinar series to answer PI's questions about grants. This was run by the NIH Pain Consortium, and it was called our Grant Mechanism Webinar Series. If you're interested in seeing these host talks, please feel free to go to the NIH Pain Consortium website. The QR code is listed here. And then another solution has been for the Purpose Network to establish a monthly Get to Know Your IC webinars. Program officers, like the ones who are here on this talk today, come on and discuss how each institute has specific efforts that they're working within the pain space. They discuss what type of pain research they fund and answer any institute-specific questions that people may have. So if you're interested in seeing any of these past recorded webinars or seeing any future webinars, I would encourage you to join the Purpose Network to be able to get access to those resources. So I've mentioned a couple of resources in my talk so far, so I just wanted to share those QR codes. The HEAL website has funding announcements, so you can please monitor that website since there may be some open announcements coming. The HEAL Workforce website, again, I'd encourage you to monitor the Workforce website to see the current and past workforce initiatives that HEAL has been working on. And I mentioned that the University of Michigan at the HEAL K manages the HEAL K-12. And so their website has the applications about if you're interested in becoming a scholar within the K-12 program. It will also share the next application window, when it will open, and there will be a webinar that explains the application processes. And then lastly, I'd encourage you to join the Purpose website if you're interested in pain research. Again, Purpose is where we really try and provide a lot of training, resources, open job announcements and really consolidate resources for everything in the pain community. It's a really good resource. So for those of you who aren't members, I would again encourage you to join. And again, Jacob will talk about in just one moment. And so last couple slides I want to share with you is another website that really is talking about all the research training and career development resources that NIH has to offer. There are a couple of websites here that are really helpful. There are a number of podcasts that both NINDS and NIH runs that helps explain how to write effective grants. There's also YouTube websites that are really useful, as well as blogs. So I encourage you to check out some of these resources to help optimize your applications. And again, please feel free to reach out to program officers. We're really here to answer your questions. And then the last slide I have for you all is that the HEAL research, the HEAL initiative is going through a strategic planning led by Jackie Ward and Lindy Wood. And the NIH HEAL initiative really wants to hear from the community. We want to get a sense of what you think is necessary for the future of the HEAL initiative. So we're really seeking public input to help shape strategic research priorities that will guide the initiative's future efforts, define the scientific solutions to the public health crises for overdose, opioid use disorder, and chronic pain. We'd encourage you to submit your feedback by July 31st, 2024. You can feel free to send an email to the email listed here and in the subject line, put in HEAL RFI and put in your recommendations. You can also go to the QR code listed below. So again, really excited for the strategic efforts and really encouraging the community to provide their thoughts for the next steps of the HEAL initiative. And so with that, I want to introduce our next speaker. Our next speaker is Jacob Coverstone. He's the PI of the Purpose Network for Pain Researchers, and he serves on the board of directors for the Alliance for Continuing Education and Health Professions and is the managing director for Neuroinnovations Education. And he's been creating professional development programs for the past 20 years. And with that, I want to hand this over to Jacob. Take it away.
Everybody. Thank you, Laura. Thank you, Anna. It's a pleasure to be with you all. As Laura mentioned, I'm Jacob. I'm the PI of Purpose. And let's see if I can control the slides. Gotta love that. All right. So Laura mentioned Purpose a bit, and you can visit by scanning this QR code or going to painresearchers.com. Our goal is to make things easier for you as researchers and for your admin teams as well. One of the ways that we do this is by centralizing a lot of community elements, in addition to preparing some exclusive content like training and webinars. Starting this year, there's new features for discussion and dissemination. And an important aspect is that Purpose is a closed network, meaning everybody, the roughly 2,900 members that are already in the network, have been vetted and approved. And this leaves discourse confidential and high level. Laura noted the funding websites that you can check out, and I encourage you to do that too. But Purpose also aggregates funding opportunities. There's a list on there from more than just NIH. We have funding notices from PCORI and CDMRP and association funding. And you'll also receive a weekly email digest of relevant information too. We host an in-person free annual meeting. The focus isn't so much on helping you to improve your research, but helping to support you as a researcher. So there's training on teamwork, collaboration, grant writing, and management. There's new elements each year. And we stream most of the sessions and record those as well. And then those are uploaded to the Purpose Media Library. And let's see here. Oh, we have tools for research admins. There are groups with discussion forums. There's also a mobile app to centralize all of this again. But we want to help reduce some of the administrative burden for admins. So in these private groups, like Rejoin, you can see an example here. They're using Purpose for their communications, scheduling all committee meetings, taking registration for their annual meeting. All of this is available at no cost. It's built right into the platform because we're trying to develop a series of tools that the entire community can use and leverage to really take things into the modern era of collaboration and dissemination. Users can discuss topics in more focused, sort of special interest groups, and then events and funding announcements and other information that would just be relevant to that sub-audience gets aggregated there as well. In our breakout group later, I can go into more depth about those things. So the real gist of this is that this is a tool. It's your tool. Users can invite other members as well. You can post information about your studies or projects. We encourage you to fill out information about your research interests, what you'd be looking for in terms of collaboration. We want you to be able to be found, find other potential collaborators, build your network, and hopefully advance collaborative science in pain research. That's really it for my talk today. Thank you for having me. I look forward to the breakout discussions at the end of the program. Turn things over to Marianne, I think. Thank you.
Thanks. Hello, everybody. I'm Marianne Pellemoner, and I lead the Pain Therapeutics Development Program, which is also part of the HEAL Initiative. Today, we are going to tell you about our program and what it offers to people who are excited about developing therapeutics to treat pain. We are also happy to have the principal investigator and co-investigator for one of our promising new projects with us today, and they will be describing their project and experiences in the world of therapeutics development from both a small business and academic perspective. With that said, I would like to introduce Dr. Matthew Rice, who is a scientific program manager for the Pain Therapeutics Development Program. He will provide an overview of the program itself, and then following his talk, Drs. John Paderi and Kennedy Goldsboro will talk about their therapeutics project. So please hold your questions until the Q&A, which is right after Dr. Paderi and Goldsboro's presentation. There will also be time for more in-depth discussions with our speakers in the virtual break room. So now I'll hand it over to Dr. Rice.
All right. Thank you, Marianne. So good afternoon, everyone. As Marianne and the slide says, I'm Matthew, and I'm a scientific program manager within the HEAL Pain Therapeutics Development Program, or the PTDP. So as you may have been aware before attending this webinar, or you certainly are aware now, the HEAL initiative is quite extensive and covers a wide range of activities: preclinical, clinical, opioid use disorder, as well as pain management. So just to provide a bit of context, the HEAL PTDP sits within the NINDS, or NINDS is the lead IC on this program, and we sit within the preclinical aspect of the enhancing pain management portion of the HEAL initiative. So why have a program like this? And I would say the short answer is that drug development, particularly in pain, is difficult. What you can see here on the lower left bar chart is just the percentage of compounds that make it to commercialization that from phase 1. So these are compounds that enter phase 1, so they've already crossed a lot of hurdles, and what percentage of that actually makes it to the market. For all diseases within the past decade, which you can see on the green bar, 6.5%. For pain, 0.7%. So not a great success rate. In the pie chart, you can see some of the reasons for these clinical failures. So unmanageable toxicity, poor drug-like properties, commercial management issues. But the big one that you see in blue there is a lack of efficacy. What you can see on the text box on the right is some of the reasons tied to this lack of efficacy, such as poor predictive validity of preclinical pain models, which the HEAL has programs to address, heterogeneous patient population within pain conditions, a lack of target engagement, predictive or patient selection biomarkers, large placebo effects in clinical trials, or therapeutic index challenges. And all of this together has really led to a decrease in the number of compounds in the pain therapeutic pipeline. So what you see on the bottom right there is from 2017 to 2022, total for both reformulation and novel entities, you have a 44% decrease in the clinical pipeline for pain management. And that is unfortunate, and it doesn't reflect the reality of the need for pain medications in this country and in the world. So that's one thing, or one of the reasons why this HEAL PTDP program exists. So to get into the program itself, so we take potential therapeutics compounds as early as hit to lead and take them all the way to phase 1 clinical trials. This is done within a U-type grant, which is a cooperative agreement, and it is quite a bit different from the R grants that I think that most people in the audience are likely most familiar with. One way this manifests, or one of the big ways this manifests, is creating this virtual pharma environment within the cooperative agreement. And this really manifests itself within the lead development team. And you can see a schematic of that on the right of the slide. So this lead development team, or LDT, is composed of the PI and their team, industry seasoned NIH provided consultants, and these consultants are tailored for the project. So if you come in earlier with example for hit to lead stage of development, we will bring on assay development consultants. If you join later in the more developmental stages, we'll have CMC, toxicology, et cetera. But the bottom line is we provide consultants with decades of industry experience tailored to the specific needs of your project. In addition to that, we have NIH staff, primarily manifesting as a scientific program manager. And then in addition, we also offer quite the selection of NIH-sponsored contracts to supplement your grant application. So within these contracts, we offer services ranging from medicinal chemistry, pharmacokinetics and toxicology, data management, manufacturing and formulation, and clinical trials. And what you'll see in the bottom right there is the funding opportunity announcement, the RFA, so you can look into that for more details. But what you'll notice there is that when looking at these NIH-sponsored contracts, it is at the decision of the PI whether to use these contracts in their grant application, whether to perform the studies in-house, or whether to subcontract them out themselves. But one thing I do want to point out is that the PI's team is always responsible for the bioactivity and efficacy study components of the application. So the PTDP itself supports biologic, small molecule, combination products, as well as formulations. We do not support device only, but again, there is a program in HEAL that does support that. These grants are milestone driven, and we'll get into a bit more detail about what that means for the structure of these cooperative agreements. And importantly, we need the PI's team to retain the intellectual property. This is a big deal for this program because the last thing we want to do is to invest a lot of time and resources into developing a therapeutic that could be helpful, only then to not be able to market it. Which is one of the issues you saw in the previous slide with that with the pie chart. So the PTDP started in 2019. Since that time, we've made 17 awards, and I'd say within the next hopefully month that will be up to 18. For these projects currently sit at hit to lead. 6 are in lead selection and lead characterization. 3 projects are either in IND enabling or preparing the paperwork to submit their IND application. 3 projects have been discontinued. So one thing that we want to emphasize is don't count on this type of grant to be the sole source of funding for your lab. And then finally, we've had 1 project that's been successfully licensed to biopharma. So if you're thinking about the research that your lab does and potentially some of the compounds you're evaluating, and you're wondering if you might be a good fit for the PTDP, again, I would encourage you to reference the RFA and read that, because that's going to have obviously a great amount of detail in it. But in general, the entry criteria, you can either enter at discovery or development. So for discovery, we want a rigorous biological rationale. We want a promising small molecule, biologic, combination device, formulation, something to that effect. and we need scientifically sound assays to optimize and test the agent. So as mentioned on the previous slide, these cooperative agreements are milestone driven, and just to provide some context, one of the milestones that are included in almost all projects are validation of in vitro and vivo assay, which touches on that third checkpoint. So if you already have a candidate therapeutic identified, you can enter at the development stage. But to qualify for this, you would have to have a very strong data package linking the therapeutic target, its modulation, and its relation to pain reduction. And you'd also have to have a strong and thorough profiling of both the biological activity and the ADME properties of the candidate that you're putting forward. So as far as the end goals for this program, again, as I said, we support its lead op. One thing we do look for is that we want the PI to suggest a target engagement biomarker that might be feasible for their compound. And again, we support IND-enabling studies all the way to phase 1. The end goals are to identify and fully characterize the lead candidate, to have that target engagement biomarker, to complete the IND and phase 1, and importantly, to seek partnerships. Because again, within HEAL, there are other programs that go up to phase 2. But at some point, if you want to get this drug to market, you will have to seek partnerships outside the NIH as the HEAL is currently structured. So again, to get back to how this differs from the R-type grant that you might be more familiar with, is that these cooperative agreements are actually broken into 2 different phases. So the first phase is the UG3, which is a preparatory phase. And that usually constitutes things such as optimization, using potency and efficacy screens if you're entering at the discovery stage, some preliminary safety toxicology data, a fairly extensive but maybe not complete profiling of the ADME properties, as well as initial optimization and PK/PD relationship between your therapeutic, target engagement, etc. At the end of this preparatory phase, you'll have a go, no-go decision that will be made. And I'm not going to get into the details of how that decision is made, but there are several bodies that are involved in making that decision. If the decision is go, you would then enter the execution phase, or the UH3, and these actually have annual go, no-go decision made each year. So for the UG3 phase, this can either be 1 or 2 years, and the total UG3, UH3 can be up to 5 years. In the UH3, we'll look at further optimization of the therapeutic candidate. We'll do more extensive, full-scale, comprehensive PK, biodistribution, toxicology. If it's a biologic, we'll do cell bank development, do other formulation work. And the goal would be for this to complete all the IND-enabling studies, and then to submit that IND and get the team prepared for the phase 1 clinical trial. So my understanding is that the audience here is rather diverse at where they are in their career stage point. So depending on where you're at, there may be a couple different opportunities to be involved in this. One is through funded research. So again, you have the RFA on screen there. We accept, biologics, small molecules, again, a range of modalities and targets. They do need to be related to pain management in some way, though. So if you feel that you have a compound that may be of interest, or perhaps you work in a lab where the PI has a compound that you think may be of interest, I would highly encourage you to have a pre-call with Dr. Marianne Pellymounter, and her contact information will be on the next slide, just to get an idea of how you may or may not fit within the program, or in what other opportunities there may be within the HEAL initiative for your compound. If you're looking at perhaps career opportunities, I would encourage you to subscribe to the listserv and get a listing of all the NINDS job announcements. Because my initial understanding is that a lot of people in this audience would have been maybe starting out in their postdoc, or perhaps halfway through, whatever halfway through means at this point. One of the job titles that you might be looking for is health program specialist. So just keep an eye out for that if that's something that may be of interest. And then finally, I'll end on this slide where you'll see the contact information for Dr. Pellymounter, as well as the other members, both within and outside of NINDS, but still NIH, that are involved in this program. You can visit in the lower left the PTDP website, which will have a link to the RFA. It will also provide more context for the structure of the program, the consultants, the contracts that we offer, and then the grants that are currently funded. So you kind of get an idea of what our portfolio looks like and what we may be looking for. In the lower right, you can watch a previous webinar that's roughly about half an hour long that goes into more detail about the program itself. And then finally, in the lowest right, you have a listing of the 2024 and 2025 receipt dates for applications to the PTDP. So with that, Marianne, can I hand it back over to you?
Absolutely. Thank you, Matthew.
Thank you.
So now it is my pleasure to introduce Dr. John Paderi and Dr. Kennedy Goldsboro. Dr. Paderi is the Chief Science Officer for IHP Therapeutics in San Carlos, California. And Dr. Goldsboro is a postdoctoral fellow at Emory University, and they're working on the same project sponsored by the Pain Therapeutics Development Program. So go ahead, John and Kennedy.
Great. Thank you, Marianne. So we are, it's a pleasure to be here and to share what we're working on with IHP. We're working on a rescue therapeutic for sickle cell pain crisis or vaso-occlusive episodes. And we're a recent recipient of the HEAL PTDP, which really has been invaluable for the development of this work. You've probably heard a lot about the exciting and really impressive science and clinical care changes with gene therapy, but I do want to make a clear point that the impact is going to be relatively small compared to the hundreds to the millions of people around the world who suffer from sickle cell, which really has just excruciatingly debilitating pain associated with it. And with that, that's really what we're trying to do at IHP is to fill in that major gap. And I'm going to hand it over to Kennedy now so that she can describe in a bit more detail some of the challenges that we have with sickle cell. Kennedy?
Yes. Thank you, John. So sickle cell disease refers to a group of inherited blood disorders or hemoglobinopathies that are associated with significant morbidity and mortality. And in the United States, about 100,000 people suffer from sickle cell disease, but worldwide, that's close to almost 8 million. And this is a disease that primarily affects people of color, specifically African, Mediterranean, Middle Eastern descent, but others as well. And the hallmark of sickle cell disease are these vaso-occlusive episodes, also known as pain crises. And these vaso-occlusive events, VOEs, are described by patients as feelings of shards of glass that are flowing throughout their bodies. And it's very severe debilitating pain, and complications from these vaso-occlusive events result in a reduction in their life expectancy that's close to 20 years. And so annually we see almost 4,000 sickle cell disease related deaths. And this is caused by the sickle sticky red blood cells that occlude blood flow to organs and their tissues. And that's really what's underlying this pain that we see. And there aren't really effective treatments for this. And so that's part of what we're working with IHP to develop. I guess I can go to the next slide too. So current treatments for sickle cell disease are primarily are heavily reliant on opioids. However, racial stigma that is still very present in this country has patients with sickle cell disease labeled as drug seekers when they're trying to get their pain treated. And we have a quote from a patient that says, "I always dress professionally when going to the ER during a vaso-occlusive event. And it's crazy that you as an African-American have to do this so you're not treated as a drug addict." In 2014, the FDA met with people in the sickle cell disease community, and the most important, the top needs that patients reported were something that they can self-administer, they can administer at home, so they don't have to go into the ER, and something that stops their pain in its tracks. And IHP 102 is something that we are working on that is unique and meets those needs.
So it's not there is some silver lining here. Sorry, we'll get to the right slide. So the silver lining in pain crisis and vaso-occlusive episodes is that this, similar to many other conditions, say the migraine with aura, or you get a bee sting and you know that you need to administer an EpiPen. Pain crisis has preceding symptoms called prodromal symptoms. And this could be like tingling fingers or an achy back. It usually is about a day or two in advance of the full-blown pain crisis, which you can see in the figure on the left. That is pain that sort of crescendos into this really debilitating and dangerous pain crisis. What that means for us is that there's an opportunity to administer a disease-modifying therapy at the beginning or the earliest symptoms of a ensuing pain crisis. And that's really what we're trying to do with IHP 102. This solves two problems. One, by the time you're in that sort of peak pain phase, that actually means that the pathologies have already occurred, the vessel occlusions have already occurred, tissue damage and organ damage is happening. The other is exactly what Kennedy described. If the last time you went to the hospital in excruciating pain, you were treated as a drug seeker, it is unlikely that you're interested in going to the hospital again. We would like to circumvent this ill-equipped healthcare system by just allowing patients to self-medicate, as we do with many other conditions, as I said, just migraine or a bee sting. And so the data that we have supporting addressing the underlying pathomechanism, recently published in Hematologica, is in this gold standard Townes sickle mouse model. What you do here is you can induce a pain crisis or a vaso-occlusive episode in these mice with mild inflammation and quantify vessel occlusions in the lung microvasculature. And what we see here is that IHP 102 reduced the vessel occlusions by more than 80%, which was really a stunning result. This is sort of the best of any therapy that has gone through preclinical development. But what is missing here is the tie between the underlying pathomechanism and pain. And that's exactly what HEAL is, at least in our beginning phases with the HEAL program, and in collaboration with Kennedy and our colleagues at Emory. That's exactly what we're tying together. So Matthew did a great job of describing the UG3, UH3, more specifically to our program. This is where we are today in year one of our UG3 phase. We had already established our molecule IHP 102. What Kennedy is working on are the acute pain studies tying together the pathomechanisms with pain behavior. And then in parallel, we're also working on our drug substance and drug product manufacture, some of the early preclinical work in safety and toxicology. And ultimately, this will culminate in a pre-IND meeting with the FDA that will then provide the roadmap for our clinical phase of development. That includes GMP manufacture, our non-clinical IND enabling studies, and then our IND filing and our phase 1 clinical trial. I'm going to pass the mic back to Kennedy because I want to have sort of hot off the press new data from our work in the acute pain studies. And Kennedy, take it away.
Absolutely. So, sorry. Recently, I've conducted a pilot study in our Townes mouse model to determine if IHP 102 does what we think it's doing. And so in our sickle mice, we're able to induce a vaso-occlusive event by exposing the mice to a hypoxic environment. And so that's the model of inducing vaso-occlusion and acute pain that we're using in my studies. And so for this study design, the mice were administered an acute injection of IHP 102. I used 30 milligrams per kilogram, which was the same dose that was used in the vessel occlusion study. And then I exposed the mice to the hypoxia chamber, which is 10% oxygen, for 3 hours. Immediately following that, they were acclimated to the von Frey testing apparatus for mechanical hypersensitivity and the cold plate test for cold hypersensitivity. And so these are two clinical endpoints that have been observed with sickle cell patients, that is mechanical allodynia and hyperalgesia, as well as increased cold sensitivity, particularly following and during a VOE. And so we thought those would be appropriate behavioral endpoints to use for these studies. And so getting to the data on the left, I tested the mice at baseline. And so prior to any exposure to hypoxia or any drug injection, we saw no difference. It's a small sample size, but no difference in the responses between the two groups at baseline. But following exposure to hypoxia, we see an increase in the number of responses that the sickle cell mice that are specifically the saline-treated mice, showing that hypoxia increases the number of responses. But we see that with IHP 102, there is a significant reduction in the number of responses that the mice experience. And so compared to saline, in the hypoxia group, we see a reduction. And even compared to the baseline values of the IHP 102 group prior to the injection or the exposure. And so overall, we see that IHP 102 reduces cold sensitivity by about 75% compared to our saline and pre-injection controls. And we also see that the observed reduction is below baseline, suggesting that this potent effect is beyond the acute vaso-occlusive pain that we see. Yeah, and I've already talked about that clinical endpoint. So we also have ongoing studies that are doing full dose responses and additional mechanism of action studies in these mice, and those are what I'm working on right now.
Thanks, Kennedy. And I just want to emphasize the point of HEAL has really been just an invaluable resource. The types of the team that they assemble, including the consultants, are really top notch, and this has been a fantastic partnership. I think we'll pass it back to Marianne.
All right. Hi, everybody. Yeah. Oh, Marianne, you want to go ahead?
No, I was just going to say let's go to Andrew. So that's great. Thank you.
All right, fantastic.
Great talk.
Sorry, jumped the gun there. So I just wanted to point out to all of our attendees here, please type your questions in the chat. We are here to answer any questions that you might have. I can go ahead and start off just having listened with some sample questions for some of our speakers. Marianne, I'm going to send this to you and Matthew, whoever wants to respond. How early in someone's career can they get involved in pain therapeutics and therapeutic development?
So Matthew, do you want to take that or do you want me to?
Well, I'll add something and then hand it over to you. I mean, I would say to be involved in pain therapeutics development, I mean, very early. I mean, you heard Laura speak to some of the opportunities that are provided. If you're thinking specifically about this program, I mean, there's no age requirement per se. The decision to fund or not will be made on the application package. So, Marianne.
Right. I mean, so I would back up exactly what Matthew said, because a lot of people start out working on pain therapeutics when they are students, when they're graduate students, sometimes even post-bac students, and then working their way through into a postdoc like Kennedy is right now. But then if you want to actually have a grant from NIH to do these pain therapeutics projects, then it's important that at least you can make it clear that you have the resources behind you to actually conduct the work that you're asking support for. So in many cases, that means that then you have a lab and you have a group that you're working with and an institution that provides the physical resources. But we don't even say specifically you have to have a PhD though, actually. So it's just resources.
Thank you very much. This is a question from Dawn Langley Brady. Any suggestions for those of us working with natural products who don't want to use animal models, or for example, if we want to use cell lines and things like that?
I can take that. So definitely there are multiple different ways of screening assets without in vivo approaches like cell and tissue-based models or three-dimensional tissue-based models or iPSC-derived models, as well as these days with primary tissues. In fact, one of the HEAL programs did develop iPSC or the pluripotent stem cell derived nociceptors cells and lines where people can use those to screen assets and look for mechanisms of action, validating target target engagement in an in vitro setup. But said all of these, when it comes to efficacy, certainly you would need an in vivo model to look for pain and pain-related outcome measures to really assess the in vivo effect, which I hope Marianne can extend that.
Right. I mean, so for some diseases and some conditions, the animal models just don't really exist well. So in that particular type of case, you still want to show some sort of a systemic physiological effect that reflects hitting the target. And so sometimes a biomarker works, but you would still need samples from a systemic type of model. So you can reduce the number of animals, but you can't completely avoid it usually. And then the other thing is when we're developing therapeutics, we also need to understand how the drug is actually getting into the body and getting out of the body so that we can better predict potential safety issues and efficacy. And so for that, we also need a whole system.
Thank you very much. That's an excellent response. I think that covered all the bases. This is a question from Rakesh, sort of talking about transitioning from looking at chemotherapy-induced pain. So I think that we broaden that question out. Are there other disease models that cause pain that can still be funded by the HEAL initiative? Is there a limit to the type of disease model that you can do if you're trying to be funded by the HEAL initiative when you're looking for pain therapeutics or addressing pain?
Matthew, do you want to take that or?
Actually, can I hand it back over to you?
Oh, okay. Yes. So one of the beauties of the HEAL initiative is that it's a collaboration between all of the institutes that are involved in pain throughout NIH. So we actually pretty much any type of pain that is a problem and is considered an unmet medical need, we can handle.
Yeah. Yeah, and I would just add, keep in mind, like the PTDP, I mean, clearly I'm partial to it, but we're not the only development program related to pain within HEAL. I mean, there are a lot, be it animal model development, so on and so forth. So.
Yes. Yeah.
And I'd just like to just quickly jump in to make just a little bit of a broader comment is that, again, I'm going to pitch the Purpose Network here. This is why we have the Get to Know Your IC webinar, is that pain is not funded by just one institute. It's funded across many institutes, and it can be really tricky to know, do you apply to NINDS? Do you apply to National Cancer Institute? Do you apply to NCCIH, right? Like there are many different institutes out there that your grant could fall into. So that is why we would encourage people to call into the grant Get to Know Your IC webinar, because we are asking the program officers to help clarify, when do you apply to NCI versus when do you apply to NINDS for some of these matters? And that might help kind of guide future application processes.
Fantastic. This is a bit of a technical question for Kennedy from Christopher Toombs, asking what the level of physical activity and core body temp is of animals in response to 3 hours of hypoxia independent of administration of IHP.
I did not measure body temperature, and anecdotally, so not in like an open field test or something, there is a slight reduction in their physical activity following hypoxia exposure, but I did not measure it, so it's not statistically. That's just what I see from the mice. Yeah.
Awesome. Thank you very much. This is from Japek Guan. Just asking, is PTDP mainly awarded as an R01 or is it awarded as other mechanisms?
So it's only awarded as a UG3, UH3. It is not awarded as an R01.
Okay. This is from Mariella Camboni. Sorry if I said that incorrectly. They had a question regarding the U-type grants, the UG3, UH3, and was wondering if the program will consider applications that include IRB-approved clinical trials like a food or supplement trial, rather than or before starting an IND-enabling study.
So yeah, I guess I can take that too. So we specifically only support trials through phase 1. And we also, Matthew described things that are coming in at development versus discovery, but both of those things are support of the drug or therapeutic discovery and development process. So we also don't support just one-off studies like a clinical trial. There are some other possibilities for if you want to run a clinical trial, and we'll have an IND, say, in 6 months, and we have an exploratory clinical trial grant program that's within NINDS. The only one of the things with that, though, is there you do have to address the mission for NINDS in terms of pain. So NINDS alone doesn't cover all types of pain. It's primarily like neuropathic, and I think DP could elaborate on that a little bit.
Yes, sure. So NINDS mission mainly includes pain associated with neurological disorders, ranging from acute to chronic pain, headache, migraine, associated with other neurological conditions like Alzheimer's disease or dementias, Parkinson's disease, spinal cord injury, TBI, and CRPS or complex regional pain syndrome. Post-stroke pain, any form of neuropathic pain where the pain condition as well as the process being studied is heavily on the nervous system. So that's what NINDS specifically covers when it comes to NINDS specific funding opportunities. As Marian and also Laura and others have pointed out, for HEAL initiative, yes, you can come with any pain condition. But for NINDS, we have to have a specific, like mission-related pain condition to NINDS.
This one is interesting. It's asking about non-HEAL pain grants. So saying things like SBIR2 grants, are any of those eligible for workforce enhancement supplements?
Yeah, that's a great question. So right now, for the specific workforce enhancement supplement that we were talking about, that is specifically for HEAL supplements. There are diversity supplements that are non-specific to HEAL. So they're HEAL-specific diversity supplements, and they're non-HEAL diversity supplements that people could apply to. So we would encourage you to look down that pathway. And again, more than happy to talk about it in the breakout sessions if you have specific questions.
Yeah, and those diversity supplements varies from each individual institute, center, and offices. So based on your pain condition or type you're focusing on, we would recommend really look into institute mission specificity and then connect to them. And the best way to start there is if you have an idea of what you are doing, just go to NIH Reporter and Matchmaker and put your summary there. It will tell you actually where it fits more, like which institute, office, or center it fits more towards.
All right, this is an interesting one. Marian, you talked about how if you already have a target in mind, that's where PDTB can be a really great program. What about grant funding for new molecules where you're searching something from scratch, like you're looking at a plant or you're trying to isolate a compound? Is there anything that HEAL supports in terms of isolating novel compounds?
So we actually, even PTDP will support, actually, we really like to support novel compounds. The question, though, is how advanced is the material for those novel compounds? And in that particular case, if it's very early and you are trying to separate out a natural product or something like that, there are some other earlier programs. There's a program led by Julia Bachman that focuses on early material and getting assays together and some of that very early tool development type work as well. So I think if you're early, you should probably, just talk to Julia, talk to DP, talk to Michael, talk to me, just to see exactly where you fit, because everything matters about the details for where you are.
Fantastic. Thank you so much, everyone, for answering these questions. I know everyone has a lot of questions that we weren't able to get to. I have written them all down, but we're going to go ahead and move on to the breakout rooms now. So I'm going to hand this back over to Anna. These are breakout rooms that are going to be with the individual speakers, and they're not going to be recorded, but she's going to have the rest of the details there, I believe.
Yes, thank you all so much. Thank you to all the speakers. Thank you for sticking around. Please do stick around for the next 20 minutes. This is your chance to meet people, see the people behind NINDS and NINDS funded and HEAL funded investigators. So please do join our breakout rooms. I'm going to post in the chat the breakout rooms by name. You'll be able to move around. So I haven't opened them yet. I will open the breakout rooms very, very soon. And you'll be able to choose which room you can join. So start in one room, move around, and we won't reconvene at the end. So stay as long as you'd like until about 2:30 Eastern, and then you're free to go. We will share the recording and slides and a whole bunch of information and contact information for all of our speakers in a week or so. All right, with that, I'm going to stop the recording and get the breakout rooms going.
Well, thank you, Anna.